This is your instructor Kwame. This lecture is on the mechanism of pathogenicity in terms of microorganisms, how they gain access to a host, get attached to specific cells or tissues, the damage in terms of the negative effects they have on the host cells, and how they leave the host cell to infect a new host. So by way of objectives, that at the end of this lecture, which is part one of a two-part lecture on pathogenicity, that you'll be able to identify the principal portals of entry and exit of microorganisms, cite some examples of how microorganisms adhere to host cells, and explain some mechanisms that they use to evade the host immune system, capsules and cell wall components, and be able to compare the effects of some enzymes that they use in penetrating the host cells, coagulases, kinases, and the likes, the functions of siderophores, the direct damage that they cause the host cells compared to the toxin that some of them produce, the effects of exotoxins and endotoxins, the different types of exotoxins, the three different types, the AB toxins, membrane disrupting toxins, and the superantigens, and be able to classify different toxins or toxin producing bacteria. In terms of terminologies, two important ones are pathogenicity and virulence. So pathogenicity is the capacity of a pathogen to cause disease. So whether a pathogen can cause a disease or not is a description of its pathogenicity. And it all comes down to whether it's able to overcome the host immune system. And then virulence is a measure of a microorganism's pathogenicity, the degree or the extent to which it can cause a disease. There are a number of factors that help microorganisms to do that. The first step would be the attachment step. So based on our understanding of the morphology of bacteria and viruses, they would use different mechanisms in terms of their attachment. And so their structural components of a bacteria, like the pili, it will produce or add adhesins or fimbriae that will help the bacteria to attach to host cells. And for viruses, if it's an envelope virus, then it will have membrane proteins that will help it with its attachment. If it doesn't, then the capsid would have structural components in terms of its proteins that will help it to attach. So to cause a disease, these are the steps that has to happen. The pathogen has to gain access to the host, be able to attach to host cells, penetrate the host cell by evading the host immune system or by overcoming it, and cause damage to the cells and tissues and be able to exit as well. So this diagram is a good pictorial depiction of what happens in terms of pathogenicity. We'll look at the first component, which is the portals of entry. So for the portals of entry, different microorganisms have different preferences in terms of which parts of the host body that they prefer entering. So some would prefer the mucous membranes, and you find mucous membranes in the conjunctiva of the eye, the respiratory tract, which is the most common portal of entry, and mainly through droplets, inhalation, or airborne pathogens being inhaled. Then there's the GI tract through food and water contaminated fingers, fecal oral transmission, and then the genital urinary tract through sexual contact. There is the skin. The skin is a very tough tissue, and so it gives us a lot of protection from microorganisms. But if there is a breach of the skin, then you may have certain organisms such as Staph aureus, which are normal microbiota on the skin that could also cause infections. Then there is the parenteral route. Parenteral route is when the pathogen is deposited directly beneath the skin or the mucous membrane, and that could be due to a trauma, through arthropods such as mosquitoes, or through injections. So the preferred sites of some organisms, for instance, if you take an organism, Salmonella typhi, which causes typhoid fever, unless it's swallowed, it doesn't cause any disease. So the preferred route has to be oral before it can produce any effects in the host. Then you have streptococci that causes pneumonia, and if they are swallowed, there is no effect. They have to be inhaled. There are certain pathogens, however, that may have more than one portal of entry. So if you take Bacillus anthracis, for instance, it could be transmitted through the skin, could be transmitted by inhalation, the respiratory system, it could be transmitted by ingestion, and so there are those exceptions. The numbers are always important. The number of organisms, microorganisms that are invading the host are important. The fewer, the better, generally. And so for most organisms, we come into contact with them, but we don't see any effects on us because the infectious dose is not adequate. The infectious dose is the dosage of the microorganism that will establish an infection in 50% of the population. And so the lower the infectious dose, the more pathogenic that particular pathogen would be, because you only need few organisms or few cells of that organism to establish an infection. Then there's lethal dose. So lethal dose is LD50, infectious dose ID50. So LD50 talks about, it's mainly in connection with toxin. So we're talking about the dosage of the toxin that will kill 50% of the test population. So if you look at that, Bacillus anthracis, Bacillus anthracis has an infectious dose that differs based on the portal of entry. So that if you look at the skin in terms of portal of entry, you only need 10 to 50 endospores. Bacillus anthracis produces endospores. The Bacillus genus generally produces endospores. And then you have, when it's inhaled, you will need a lot more, 10 to 20,000 endospores. When it's ingested, you need actually a lot more, 250,000 to 1 million endospores in order to establish an infection. There is this article here that you should look through. It gives more information about Bacillus anthracis, the different types of the disease in terms of the portal of entry and how it presents. Then after numbers, another factor that is important for pathogenicity is adherence. So most pathogens would produce adhesins. So adhesins are surface projections that you will find on the surface of these pathogens. They are mainly glycoproteins or lipoproteins. Glycoproteins, you're talking about sugar and protein combinations, and lipoprotein, you're talking about lipids and protein combinations on the surface of the pathogens. And it is with these molecules that they're able to form complementary receptors with the host cells. So it becomes like a lock and key. So this is a host cell, and this is a pathogen, and these adhesins here are able to form complementary bonding with the surface proteins or the glycoproteins of the host cells. So if you take Streptococcus mutans, for instance, it will produce glycocalyx adhesins. E. coli would have fimbriae, pili, flagella. That also helps it in attaching to host cells. Host cell receptors, in most host cell receptors are mostly sugars or combinations of it. So you may have a glycoprotein, which is sugar and protein combined. And these are the attachment to which the pathogens will adhere to. Bioforms also help pathogens to attach to host cells. And with bioforms, it gives them more resistance as well to treatment of antimicrobial drugs. The second component, so after the organism has gained entry, has the right number, and is able to adhere to the specific cells, it should be able to either penetrate the host defense or evade it, either overcome it or evade it. And there are a number of mechanisms that the cells of these pathogens would use. They may use capsules. Capsules will prevent or stop the process of phagocytosis. Some may have cell wall proteins, such as M protein in Strep pyogenes, which will also help it to evade pathogenesis. Others will exhibit what we call antigenic variation. Antigenic variation helps these microorganisms to avoid the immune system. And if you take organisms in the genus Trypanosoma and Neisseria, they exhibit this particular characteristic. And all that it means is that the pathogen is able to change the proteins on the surface, the proteins or carbohydrates or combinations of proteins and carbohydrates on its surface, just in time, quick enough, so that by the time the host cells mount a defense, the pathogens has already changed its antigens, its surface proteins, so that the antibodies that are produced by the host cells becomes useless. There is a new antigen. And so this graph tries to illustrate that. So it has these antigenic proteins at this stage, and as time goes on, it changes to different antigens, it changes to a different one again, and different again. So it's always one step ahead of the immune system of the host. So if we're looking at capsules, how capsule is used to evade the host defenses, what it does mainly is that it prevents the activation of complement proteins. Complement proteins are one component of our immune system that helps in fighting foreign organisms. So cells, pathogens that have capsules, for instance, wouldn't have their C3 B receptors open for them to attach to the white blood cells and to activate that component of the immune system. So they're able to escape phagocytosis. Examples of such organisms would be Strep pneumoniae, Klebsiella pneumoniae, and Bacillus anthracis. Then in terms of the cell wall component, you have the M proteins that are present in, for instance, Strep pyogenes. M proteins are found in the cell wall. They are heat resistant and acid resistant as well. But the most important thing that they do in terms of helping the organism to overcome the host immunity is that it helps it to attach, so immediate attachment, but it helps it to evade phagocytosis. So this is the structural arrangement of M protein, and in there it has a components that help it escape the immunoglobulin A action, so it inhibits the action of IgA. And then in addition to that, it has these proteins, C4BP, which also prevents phagocytosis from occurring. We've noticed that organisms that do not have the M proteins in their cell wall, those are mutants. Those mutants are susceptible to phagocytosis, so they're not able to overcome that. Other microorganisms such as Neisseria gonorrhoeae would have proteins in the cell wall such as OPA protein. Organisms that have OPA protein, when you grow them in a culture, they appear opaque. But OPA protein helps such organisms that have them to attach easily to the host cells, and then they are easily then internalized, meaning using the process of endocytosis, they are easily able to enter the cell through endocytosis. They are able to outcompete the microbiota that are present on the cell because they're able to attach and multiply, and then easily transported into the cell by endocytosis. Then you have organisms like Mycobacterium tuberculosis, which causes tuberculosis, which has mycolic acid, the acid-fast organisms. Now, with the mycolic acid, what it does is that they're able to avoid phagocytosis and even multiply within cells that are supposed to destroy them, the phagocytes. So that is also another mechanism that some organisms use. If you look at organisms like Salmonella and Listeria, they produce proteins or enzymes known as invasins. So you're looking at the word invasion, so it helps them invade the host cells. And what these do is that they attack the host cell cytoskeleton. They attack structural proteins of the cell. Salmonella, for instance, would produce these invasions, and what the invasions will do is that they will disrupt the cell membrane of the host cell and create an effect, as you would see. For instance, if you have a drop of water drop on a solid surface, it just splashes, and what the protein does is similar to that. So this effect is called the ruffling of the host cell plasma membrane. It ruffles it and makes it easier for the Salmonella to enter the host cells. Others, such as Listeria, will produce actin that will help them, that once they're able to enter the cell, they're able to move easily to other cells, spread easily. Other microorganisms will produce enzymes that are released extracellularly, so they produce the enzyme and they release into the environment. If you take Staph aureus, they will produce coagulase, and what coagulase will do is that it will form a clot around the microorganism and prevent it from being destroyed by phagocytes, and that makes it virulent. Others would have kinase, like streptokinase, that are produced in Streptococcus bacteria, and what they do is that they dissolve clots that are formed. So when there is a breach in the skin, pathogens are rounded up the cell, immune system is able to identify them, and the clot is formed around it, and it's being readied for destruction. These pathogens that produce the kinase enzymes will dissolve the clots and continue spreading in the host cells. Others will have hyaluronidase, which more or less breaks up the component of the cell that helps attach cells to each other. We call it cell cement, the cement of the cell, and so it loosens things up and makes it easier for them to spread intracellularly. The others have collagenase, which are also structural proteins that are found in connective tissues such as the skin and the cartilage, and they will break these down. Others would break down immunoglobulin proteins, and that prevents the immune system from destroying them. So by way of some depiction, this is how coagulase work generally. It will form a clot around a microorganism, and therefore phagocytes are not able to destroy them. But the opposite is true for streptokinase. So these kinase enzymes, what they do is that once the cell of the host immune system forms a clot, if there is a breach, it will form a clot around potential pathogens. But if they produce kinases, they will then dissolve the clot and then enter the bloodstream and continue to spread. Those that produce the hyaluronidase will be able to cause the cells to be more loosely attached than they usually are, and thus offer an entry point for them to enter the cells, other cells of the organism, or the intracellular components of the organism. The next step after the organism has gained entry, has the right numbers, has been able to adhere, been able to overcome the immune system, is that it's going to cause damage to the host cells, or damage the host cells. And there are a number of ways that organisms do that. They could use the host nutrients, they could cause direct damage, they could produce toxins, or they may induce hypersensitivity, meaning the immune system response to the pathogen is an exaggerated response, and that in itself becomes a problem for the host. So we'll look at just how they use the host nutrients and direct damage that it cause, and continue with a toxin aspect of the host cell damage in the next lecture. Now, in terms of how they use the host nutrients, the pathogens generally, or most microorganisms, will need iron for growth. So most of them will need iron for growth. Iron, however, has to be free and loose for them to use. But in most hosts or organisms like humans, ions are not free. Ions are bound to certain proteins. So you have proteins like lactoferrin, transferrin, ferritin, and hemoglobin that bind to iron. Now, for the microorganism to get access to these ions, they should be able to, one, outcompete the host cells for the ion, and sometimes they do that. So there are certain microorganisms that have special mechanisms called siderophores. So these siderophores are some structural components that they have, which help them take away the ion from the host cells by binding more strongly to the ion than the host cell proteins would do, and therefore they're able to compete them. And so that's one way of doing that. There are other mechanisms that are employed after they bind to it. They may just release the ion into their cells directly. They may form a complex with it. So when a complex is formed between the siderophore and the ion, the two may together enter the pathogen cells. There are other mechanisms as well where there would be a direct interaction with the host ion transport protein. So for instance, if hemoglobin is bound to ion, the pathogen may also attach its siderophore to it, so that you end up having an ion transport protein and siderophore complex, all three of them. And then the three are then sent, or using endocytosis, are then introduced into the pathogen cell. Another way would be for them to destroy the host cells and release the ions, and then be able to incorporate them into their cells and use them for growth. So there are a number of ways that siderophores are used. Cells may also be destroyed, host cells may also be destroyed directly by the actions of the pathogen. And this also happens more common. For instance, if you have an organism that is resistant to phagocytosis, then in the presence of phagocytes, it will still continue to multiply. And as it multiplies, it's going to use nutrients from the host cells, and in the process will produce waste or byproducts. And these may cause direct damage to the host cells. Then there's the issue of hypersensitivity, which would also result in damaging of host cells. So this brings us to the end of part 1 of our lecture on microbial pathogenicity.